Hi, I'm Navya. So, I'll be taking a class for the physiology subject, that is for the second year students of the physiology that comes under the paramedical course. And the chapter name is second. So, that is the blood. So, let us know about this chapter in today's video. So, the blood. So, this blood is the fluid connective tissue. So, that consists of the plasma and RBC and WBC, that is red blood cell and the white blood cell and the platelets. These four. So, let us know about the plasma. So, this plasma is a main component of the blood, and that consists of mostly of the water and also with this protein and ions and the nutrients, also the waste mixed with that. And in RBC, that is red blood cells, so those are very responsible for carrying of the oxygen and the carbon dioxide. And the platelets, so these are very responsible for the blood clotting. And WBC, that is a white blood cell, so these are the part of the immune system, and also it function in the development and in the immune responses. And the next concept is that composition and function of the blood. Composition. So here the cells and the platelets. So they are both made up of the 45% of the human blood, sorry. So while this plasma, it is made up of other 55%. Composition is nothing but the divisions of the percent has been made up with the plasma as well as the um arthrocytes. So here, and remaining one, it will be less than 1%. That will be the buffer code, that is leukocytes and the platelets. The total amount of the blood it consists of. And in the plasma, how much it is? Yeah, 55% of the plasma. So let us know about this plasma, the composition of this plasma. So this plasma it will be in the liquid state that can be contributed to the plasma, that and it makes up like 55% of the blood. So this will be in the pale yellow color. Then when it is separated, it consists of the salt, nutrients, and the water enzymes. This blood plasma, this is also contains of the important protein along with this and other component that is very necessary for the overall of the health. And the platelets. So here, this is like a tiny disc shaped cells. So that helps in the regulation of the blood flow. So when any part of the body is in the damaged condition, then this aiding is very fast. I mean, the recovery stage will be very fast through the clotting of the blood. Totally, we can just tell it about the easy recovery. Easily, the patient can recover through this clotting of the blood. And RBC. This RBC, this is also known as the erythrocytes. So here in this RBC, this contains mostly this iron-rich protein that is called as a hemoglobin. Iron-rich proteins, these are called as a hemoglobin. That gives color. I mean, that gives the blood it's the red color because of the presence of the hemoglobin that is rich in the iron, rich protein. And the main function of this is to transport the oxygen from and to various of the tissues and the organs. The RBC, and we have the WBC, leukocytes. So these are colorless blood, white blood cells. So these are the colorless blood cells. So they are colorless, we know, because it is devoid of the hemoglobin. And we have a types, that is granulocytes. So that types. So granulocytes is nothing but they are the leukocytes. Leukocytes comes under WBC. So within the presence of the granules in the cytoplasm, so these granules, granulated cells, so that include the basophil and neutrophil and eosinophil. So here granulocytes, these are the, again it comes under the same division, that is the leukocytes. Leukocytes is nothing but the colorless blood, WBC. So this will be with the presence of the granule in the cytoplasm, the in the cytoplasm. And aggregates, these are again, it is again divided into monocytes and the lymphocytes. So this lymphocytes, let us know the lymphocytes. So this lymphocytes, this plays a vital role in the producing of the antibodies, and the range will be 8 to 10 micrometers. So these are commonly known as the natural killer cells, and also they play a very important role in the body defense also, protects the body. And these white blood cells, those are colorless cells, we know this, and it is formed in the lymphocyte tissue, and this is also referred to as a lymphocytes. Why it is called as also the lymphocytes is again, but it is formed with the lymphocyte tissue. So there are two main types of this lymphocytes, that is a B lymphocytes and the T lymphocytes. B and T lymphocytes. Lymphocytes is nothing but the WBC. So here, yeah, as we already discussed, under this blood, 55% will belong to the plasma and 45% belong to the blood cell. So under the blood cells, the red cells and the white and the platelets comes under here. Already we have learned that red blood cells is help in the transportation of the oxygen. White is for the defense purpose, and the platelets, when it is got when the body, when the part of the body got damaged, it just recover easily, that is of the clotting of the blood. And phagocytes also we have, and lymphocytes also we have under this white cell. Phagocytes, this helps in the ingestion of these microorganisms, and also in lymphocytes we saw B and T cell types, that is a B cells and the T cells. This B cells that produce antibodies to the body, and the T cells that will kill the infected and the cancerous cells in the body. And in the plasma, that is a water, it consists of almost 92% of the water, and the dissolved substance, that is 8%. So under this dissolved substance, that may be the hormones and the food that may be dissolved, and the salt, 3.5% of the salt. In the food, it contains like glucose, amino acids, and the fats, these types, and also the protein and the waste products, that may be the carbon dioxide and the urea, the waste product. Under this protein, again we have a two division, that is a fibrinogen and the and also this fibrinogen, this is for the clotting, and albumin, this is for the maintenance of the osmotic pressure of the blood, it maintains that pressure. And now let us know what the function of the blood includes. So usually common function is that it supplies the oxygen and the carbon dioxide. So between, where does it supply? Between the lungs. So it supply between the lungs and rest of the body, and the rest of the body. And also it supplies the essential nutrients. It supplies the oxygen, carbon dioxide, and also it applies the essential nutrients to the cells, such as the amino acid. So I'll just mention here: carbon dioxide, oxygen, amino acids, fatty acids, fatty acids, and the glucose. And it's transports the waste products, so that may be to be removed by kidney. The waste products are removed by the kidney, and it transports the hormones, and it has a role in the inflammation, that is an leukocyte, so that destroys the microorganism. That platelets also initiate the blood clotting, etc. So here the function, it supplies oxygen, carbon dioxide, and essential nutrients, that is amino acid, fatty acid, and the waste product that is removed from by the kidney, and the hormones, and the glucose. So and also it provides, it has a role, that is inflammation. These are the function of the blood. And also the body temperature, it just regulates the body temperature and also the pH level, acidic or the bases. And now we have the plasma. So under this plasma, the composition and the function of the plasma proteins that you're going to learn here. The composition, in the case it is a division, and the function of the plasma. So this plasma, this is also known as a blood plasma, and this appears in the color that is a yellow, light yellowish color, or the straw colored just carried, and it serves as a the liquid base. It serves as a liquid base for all blood. And here RBC and WBC, the platelets, these made up of all the plasma. So uh, this is formed from the water and the salts. For water and the salts that will be absorbed through the digestive tract. This plasma contains 91% or to 92% of the water and 8 to 9 of the solid. So 91 to 92% of the water and about the solid. And it comprises of the coagulants, that is mainly fibrinogen, and that aid the blood clotting. Then the plasma proteins such as like albumin and the globulin, so that helps in the maintenance of the colloidal osmotic pressure at about 25 mmHg. And these electrolytes like the sodium or the potassium or the bicarbonate, chloride, and the calcium, that helps in the maintaining of the blood pH level. And immunoglobulins, that helps in the fight infection, and also other amount of the enzymes and the hormones and also the vitamins. And let us know the function of the plasma proteins. So this forms a liquid base of the blood. So the function that may be carried out by the plasma and the blood overlaps, in that means that it has a multiple function, that is a coagulation. So here fibrinogen, that plays a major role in the blood clotting, and also along with that other coagulants like the thrombin and the factor X. And also it is for the defense purpose, that is immunoglobulins and other antibodies that in the plasma, that plays a very important role in the body defense against the bacteria. Defense in the sense against the bacteria, virus, fungi, and the parasites. It will um, act as a defense purpose. Use And also we have the maintenance of osmotic pressure. So here the colloidal osmotic pressure, that is maintained at around 25 mmHg by the plasma proteins like the albumin. And also we have a nutrition, that is the transportation of the nutrients like the glucose, amino acids, and lipids and vitamins, and that will be absorbed from the digestive tract, that will be to different parts of the body as a source of the fuel or the growth and the development. So these are the function of the plasma proteins. And now let us know the other concept of the cellular components of the blood. So under this cellular components, here we have RBC, WBC, platelets, and morphology, the naming convention. So here the cellular components of the blood, that includes red corpuscles, and also we have the 5 types of the white corpuscles. We used to learn this red corpuscles and 5 types of the white corpuscles in the platelets, and the platelets, that is thrombocytes. So the shape, in the sense, it is like tiny disc-shaped cells. So that helps in the regulation of the blood flow when any part of the body that is in the case that is damaged. Thereby, this aiding is a very fast recovery. The platelets, so very fast recovery. We can find this because of the clotting of the blood, that is through the platelets. And let us know the difference between the red blood cells and the white blood cells. So red blood cells, so these are called erythrocytes, but here in the white blood cell, these are called as mucocytes or leukocytes. And other difference, RBC, those have biconvex disc shape, but here this may be irregular shape. And the size of this RBC, that varies from 6 to 8 micrometer, that is in the diameter, but here it varies 12 to 17, that is also major difference. And the lifespan of this RBC is about 120 days. but here this will be very less, that may be extend for 12 to 20 days. And RBC, the cells do not have the nucleus on maturity. On the maturity, it doesn't have any nucleus, but here, so these RBC, these will be categorized by the presence of the large central nucleus. And due to the presence of this hemoglobin, the color will be in the red in the RBC, whereas here it is a colorless and do not have any pigment. Like in the case of a red red blood cells, here it doesn't have any pigment, it will be in a colorless. So here in an RBC, it's only one type of RBC is will be exist. But in the case of WBC, so different types of WBC are found in the blood. So such as the neutrophils and B lymphocytes and T lymphocytes and monocytes and basolines. So these are the different types of WBC has been found. And also RBC, this helps in the transportation of the respiratory gases to different parts of the human body, but here it is also helps in the producing of the antibodies that is used to fight against with the disease that causing microbes. And here RBC, these are produced in the red bone marrow, but this also produced in the red bone marrow. Also we have the lymph node and the sleeve we have. This components of the red blood cells or the hemoglobin, because the color of this red blood cells in the red color, that is due to the presence of the hemoglobin, whereas WBC is colorless. So the component of WBC cells are antibodies that will be with the presence of the MHC, that is a major histocompatibility complex antigen cell markers. And here in RBC, so these cells that make up around 36 to 50% of the human blood, but here that will be around 1% of the human blood. And next concept is the formation of the hemopoietic cells. The formation. The hemopoietic stem cells, these are the stem cells. So what does it produce? That give rise to the other blood cells. So that process is called as a hematopoiesis. And here in this process, this occurs in the again it occurs in the red bone marrow, that is in the core of the most bones. And in the embryonic development, this red bone marrow, what does it will do? Means this will be derived from the layer of the embryo that is called as a mesoderm. This hematopoiesis, this is a process by which all the mature blood cells, those are produced at the same time, and it must be balanced enormously to the production cases the needs. Hematopoietic stem the cells give rise to the different types of the blood cells, and that is also called as the myeloid or the lymphoid. Myeloid and lymphoid, these are both are involved in the dendritic cell formation. And this myeloid, in the case, it includes the monocytes, macrophages, neutrophils, and basophils. Whereas this lymphocells, that includes the T cells and the B cells, and also the natural killer cells, and in each lymphoid the cells. And the lifespan. The next concept is a lifespan. So here, this lifespan, I mean here the details in analysis of this large panel of the HSC, that is shown in the lifespan of the individual HSC, the individual cells, that is with the 10 to nearly 60 months it has been given, and that will be found in the under the side by side in the same total, that indicates maybe lifespan of the predetermined on the level of the HSC. There's a hemoglobin. And next topic, that is the hemoglobin, the formation and the function and the destruction of the RBC, that is the applied aspects of the hemoglobin. So now let us know one by one the hemoglobin. This hemoglobin, this is also spelled as the hemoglobin. This is the iron containing protein in the blood that will be present in the blood of the many animals, that will be the red color. It's a red blood cell of the vertebrates that transport oxygen to the all the means to the tissues. This hemoglobin, this forms an unstable, it is a reversible bond with the oxygen, and in the oxygenated state, this is also called as oxyhemoglobin. only in the oxygenated state, and is very bright in the color, that is a red, and that is a reduced state when it is in a purplish blue. Hemoglobin that develops in the cells in the bone marrow, and that become a red blood cell. So it first develop in the bone marrow. And next, let us do the function of this hemoglobin. This hemoglobin has a multiple function. The first thing is that it transports the oxygen from the lungs to the tissues, from the lungs to the tissues. So that facilitate this phosphorylation that is the mitochondria. And also, first thing is it supply oxygen from the lungs to the tissue, and also it carriage of the carbon dioxide, with the carriage of the carbon dioxide from the tissue to the lungs. So it transport oxygen from the lungs to the tissue, and it carries back to the carbon dioxide from the tissue to the lungs back. And the buffering of this hydrogen ions that is formed in the atropine from the conversion of the carbon dioxide into bicarbonate. And the destruction, let us know the destruction. These are removed by the reticule endothelial system, and also the globin chain will be there. So those are broken down to the amino acid, so which will be returned to the amino acid pool. This iron are reused by the bone marrow, that is used for the synthesize purpose, and also this will be extracted by the bile, and that will be into the small bowel. And in the gastrointestinal tract, the bilirubin that will be converted into the stercobilin, and some is reabsorbed into the plasma also, and those are excreted by the kidney as urobilinogen in the urine form, and the small amount of this free hemoglobin that may be released into the plasma. So, and also the concepts we have, that is ESR, PCV, blood indices, and the anemia. So ESR. So this ESR, let us know this concept first. So ESR, this is the measurement of the rate of the sedimentation of the red cells, sedimentation of the red cell, that is in the anticoagulated blood. So in the anticoagulated blood, that measures the sedimentation of the red cells. So this blood is allowed to stand for 1 hour. Just 1 hour it will stand, and that is in the open-ended glass tube only. That will be mounted vertically on the stand. That is on the stand, that is length of the column of the plasma that is above the red cells. And the PCV, that is a packed cell volume. So this is the measurement of the proportion of the blood that is made up of the cells, and the value is expressed as a percentage or the fraction of cells in the blood. For example, fraction in the case, a percentage and fraction in the case like PCV, that is packed cell volume, that is 40%, means that there are 40 milliliters of the cells in 100 milliliters of the blood. So 40 milliliter of the cells in 100 milliliter of the blood. So that indicates, and the blood indices. So here the blood indices, that is the red blood cells. So that indices are the blood test. So it is something with the blood test that okay, why do the patient go for the blood test? To get information about the disease. Yes, so to provide information about the hemoglobin content. So what kind of the bacteria infection has been contained in the blood. So that's why for the test, blood test, because that provides the information about the hemoglobin content and the size of the red blood cell. And this abnormal values that also indicates the presence of the anemia, and which the type of the anemia that is included. So MCV, that is a mean corpuscular volume, that is the average volume of the red blood cell, and that will be calculated by dividing the hemocrit, that by the concentration of the red blood cells. By this is a formula MCV, that is a hematocrit, that is divided by RBC. The normal range will be 80 to 100 FL, that is a femtoliter. This mean corpuscular, this hemoglobin, that is the average amount of the hemoglobin per red blood cell, that is calculated by dividing the hemoglobin by the red blood cells count. Yeah, dividing the hemoglobin by the red blood cells count, that will be 27 to 31 pg per the cells. And the next concept is the blood group. Yes, this is some more interesting topic. Blood group. So here we have a different types of blood group. That is the ABO blood grouping method we have, and also the RH typing, a Land's-Strange law, cross matching concept, storage of the blood, the indication and the contraindications of the blood transfusion. So these are the concepts we use to learn under the blood grouping. So first learn about what are this blood group system. So the blood grouping system, this is a pivotal in the blood transfusion. It's very important when the patient is going to transfer the, just he has to donate the blood to the some beneficiary person. When the person is suffering any disease or any damaged parts, so then this transfusion, it plays very important role. So the immune system, so that recognize another blood type that as a foreign or that attacks it if it is introduced in the body, that is causes a transfusion reaction. So why in the case is if any inappropriate match happens with the RH, that RH and ABO blood types, that may cause very serious issue to the person. If the blood, if the patient of the blood doesn't matched, then definitely it will be a serious cause. So therefore, this blood transfusion is very important while checking before the blood is transferred before from the donors to the patient. So what are these ABO and RH blood group? So what happened? So during the blood transfusion, the two most important group system will come into the picture. So those are the ABO system and the razor system. This ABO blood group system, so that consists of, again it consists of the four types. ABO, it consists of the four types, that is A, B and AB and O. These are the four types. Under this ABO, it comes in the picture. So this is mainly based on the antigens. So this four types is based on the antigens and the antibodies, that is on the RBC and in the plasma structure also. So this both antigens and antibodies, those are the proteins molecules. So what happens? So that antigens will be present on the surface of the RBC. So both will be present actually. So antigens and the antibodies. So antibodies also present in the plasma. So in the both are present in the plasma as well as the RBC. So this RH blood grouping system, so this consists of the 50 defined blood group. So 50 defined blood group of the antigens. Under this RH system, now we are talking about RH system. So the most important antigens, those are the T, C, C, E, E. So that's it. That's why this ABO and RH blood system, those are discussed in the below. So that is about, yeah. So now let us continue the same thing about RBC blood group system. As I told you, there are four blood groups, that is a group A, B, AB and O. So here it consists of the two antigens, that is antigen A and antigen B. Here ABO grouping system, so that is classified by the four types based on the presence or the absence of the antigens, that antigens on the RBC surface and the plasma. So that should be present in RBC as well as the plasma. Yes. So here, let us know about the A. So that contains antigen A and antibody B. So as I said, it consists of the antigen and the antibody. Antibody. Here. So A consists of the antigen A, antibody B. In the B consists of same antigen B, antibody A. AB. So here it contains both A and B antigen, and there will be no antibody, not either A, not either neither A nor neither B. And O. So here it contains neither A antigen or neither B antigen, but both antibodies will be present. This both are like vice versa. It contains both antigen A, B, but not and also both antibodies A and B. It neither contains this, but it contains this. Just remember this. Group O, it's neither contain A or B antigen, but here it contains both antibodies, that is A and B. Yes, so now this is ABO group system. So this is very important during the blood donors or the blood transfusion. When the person, when the patient is coming for to get needed of the blood, that this is very important, and for the donors also this is very important, because in any case the mismatching of the blood, that may cause a very serious issue for the patient, and it's very important for the blood cells to match. Otherwise, it will be very serious issues for the person. For example, if the person of the blood group of A can receive blood either from the group A or the group O, as there are no antibodies. That's why, as there are no antibodies, so he can receive, otherwise could not. Now, Rh blood group system. So in addition to this ABO, there are the outer prominent one, that is the Rh groups in grouping system also is there. So here about two-thirds of the population that contains the third antigen on the surface of their red blood cells, that is known as a Rh factor. This is not for the all, but it's like two-third people, two-third population that may contains of this, and this decides whether the blood group is positive or the negative. Rh factor only decides that the patient is a positive or the negative. If this Rh factor is present, then the individual is a rhesus positive. If Rh factor is absent, then it is a negative. We used to tell the positive and the negative. So Rh positive, Rh negative. If yeah, this is going to judge in this case like this. If Rh factor is present, then it is positive. If Rh factor is absent, then it is negative. That is how. Yeah, this is somewhat the classification of this group A, B, AB, and O. So here, anti-B antigen is A, the presence of here antigen, and anti and antigens are present here. And the next is the Lansky's law. So this is a reciprocal relationship between the antigens on the red blood cell and antibodies in the serum. So antigens and antibodies, these are important. So that is known as a Lansky law. So it says that if an nitrogen is present in the RBC, then the corresponding agglutinin that must be present in the plasma. And also we know the application. So the first law holds true for all the three of the blood grouping, and the second is if it's in fact for the ABO blood group, and also RH, MN, and other blood groups also doesn't follow the second part of the Lansky law. And the cross matching. So this cross matching is nothing but the testing before the blood transfusion. Just they're gonna check whether it is going to match or not. That is to determine if the donor blood is compatible with the blood of the intended recipient, the person who needs the blood. So they used to cross check it. And this cross checking, this is also used to determine the compatibility between the donor as well as the organ transplantation. This compatibility, once if it is done well, then they gonna ahead. Otherwise, they just stop it. So the compatibility, this is determined through the matching of the different blood group system. The most common is that ABO and the RH system. And this is directly testing for the presence of the antibodies against the antigens. They just check about the antibodies in the presence of the antigens. This cross matching is done by the certified laboratory, not in all. The certified laboratory, it can be done there, and the technologists with the in front of the technologist and in the medical laboratory. And the next concept is the storage of the blood. Yes, this is very important, because if in case improper storage of the blood, then definitely it is a loss of that blood. It may not be reused, it may not be used to any other patient. So the storage of the blood is very important once the blood is collected from the donors. So usually the blood is collected into the plastic bag. We all know this. The plastic bag they used to collect from the donors, and they used to store that which used to contain with the anticoagulant or the anticoagulant that which allows, so that allows it to store for some days, sorry, some hours. And so that includes citrate phosphorus dextrose, citrate phosphorus dextrose, and the acid citrate dextrose, ACD, or so it is commonly known as ACD or CDP, that is with the adenine. So that to prolong for the red cell storage, and other preservative solutions also like SAGM. This is why they used to these why they use all these things is to just for the safe storage, and it should not be get spoiled. And if the red cells are preserved in this, if the red cells are present in SAGM solution, then the lifespan may differ. It will go up to 42 days. The SAGM, if it is stored under SAGM, then the lifespan will be 42 days. In case of this, so I'll just write up here. Yes, CGM, that is for the 42 days. In case CPD, if they preserve under this, so here that will be stay for the 21 days, whereas the solution preserves for the blood, that is also for the 35, 21 to 35. Here the whole blood, but here it is for the blood for 35 days. And the material of the bag is the bag which contains of the blood, that is, so the bag, so that is bicompatible. So the bag is bicompatible with the blood cells, and also it allows diffusion of the gases, permitting optimal cell preservation. It just preserve it. And the specimen that's collected is tested for the blood, so which they have collected, that will be tested for the hepatitis B or the C and HIV. These are the main disease they used to check for the, because they used to give it to the patient, so they used to check, they used to test all these type of tests they're gonna take up: hepatitis B, hepatitis C, HIV, and other infection they used to check up before giving this to the, before injecting this to the patient. And the blood is stored then in the refrigerator, so that is under 2 to 6 degree they're gonna store. And each unit of the whole blood, that is normally they're going to separate it into the several components. This red blood cells may be stored under the refrigerator for the maximum of the 42 days in the refrigerator. Then they be frozen up to for the 10 years, maybe frozen also up to 10 years. This red blood cells also carry oxygen, and also it is used to treat other anemia. So the blood bank, so what is a blood bank? Is something, it's a center. So we used to get the blood, right? Yes. So here, where the blood is gathered, as a result, the blood donation is stored there, and also it is preserved for the later use of the blood transfusion. And here, the T bank. So those typically refer to the division of the hospital. Means they have been divided for all the hospital, east, west, north, south direction, and there has to use to where the storage of the blood, that products occurs where the, means when there is a availability, when there is a necessary, here there will be the availability. Yeah. And next is the indications and contraindications of the blood transfusion is there. So here the blood should be treated as the drug, right? Just to storage, otherwise properly, otherwise it may get infected also. Yes, so that must be preserved with very caution. So there are both indications and contraindications before is there in giving the blood transfusion. Indications are that includes the severe anemia. So here, this severe anemia, when the oxygen capacity of the blood comprises of major organs, this happens. And the severe hemorrhage and anemia of the chronic disorders, in the chronic in the sense the renal failure and the cancer, kidney cancer. So these type of issues are being present. And hemoglobinopathies, there is a sickle cell disease and thalassemia. And the contraindication that includes megaloblastic anemia, that is a vitamin B12 or folate deficiency, and that will be the transfusion that may cause a heart failure and sometimes death also. And also the major thing is the iron deficiency, that is the anemia, and the transfusion in the healthy adults and the children way use of the oral iron, and that will be very low hemoglobin. And the next concept is a hemostasis, that is a clotting factors, anticoagulant factors. Yeah. So hemostasis. This hemostasis, this is also spelled as a hemostasis. So this is a process that is to prevent or to stop bleeding. So the meaning is to just keep the blood within a damaged blood vessels, not to out of it. Yes, so this is the first stage of the wound healing. If you stop the blood from this, then it will be like healing stage only. This is a prevention thing, means overflowing of the blood. So this involves coagulation and the blood changing from a liquid to the gel. I mean the stage. So liquid, it is a change. The blood will be changing from the liquid to the gel when any wound, any cut has happened to the body. So this has a three major step, that is constrictions and the temporary blockage of the break by the platelets plug, and also the third one is a blood coagulation or the formation of the fibrin clot, and also we have the clotting factors. This coagulation, this is also known as the clotting. So this is a process by which the blood changes from the liquid state to the gel state. That is the clotting factors. So that forms the blood clot. So this is potentially that results in the hemostasis. So, it's nothing but the changing from the liquid to the gel. And the following factors that increase the risk of developing the blood clot. That is obesity, the main thing, and the pregnancy, immobility, and the oral contraceptives, and certain cancers and trauma, certain surgeries. These are the it has been involved smoking also. Anticoagulants. So the next concept, anticoagulants, these are the medicines. So what does it do? It just prevents the blood from the clotting. So it just acts like a prevention from the clotting, and also it quickly acts as effective for the normal. Anticoagulants, these are used to treat and to prevent the blood clot that may also occur in the blood vessels. So this blood clot, this blood clot, this can block the blood vessels. Anticoagulants, this is commonly known as the blood thinners. So those are the chemical substances that prevent or reduce the coagulation of the blood. And some of them that occurs very naturally, that is a blood-eating animals such as the leeches. Yeah. So this purifies the blood, and the mosquitoes, not and unclotted along with the enough animal that will obtain some blood. Bleeding time and clotting time, this is very important, because when the patient get so that will be affected in the infected or damaged or any wound, any cut, so then this bleeding time will it matters a lot. Or any the person in any case, like the accident case, the bleeding time is very important to be noted down, and within the bleeding time the patient or the person has to be medicated, otherwise it will be very it will be in the serious condition. So bleeding time, this is a medical test done on someone to access the platelet function. So this involves making patient bleed that timing. So how long it takes them to stop the bleeding. This is very important. So the term template bleeding time, this is used when the test is performed to the standardized parameter. And the normal bleeding time, that will be between 2 to 7 minutes. This is like normal. Nothing will happen to that person who is getting so who will be bleeding for 2 to 7 minutes in the case of wound or any cut on the body. And the clotting time. So this time is a time that is required for the sample of the blood to coagulate in the vitro under the standard condition. So that conditions are that maybe there are like various types of determining the clotting time. The most common thing, that is a capillary tube method. So this is affected by the calcium, iron levels, and many other diseases. And normally normal value of this clothing time, that will be 2 to 7, so 2 to 8 minutes, that is a normal thing. And the normal clothing times in a person between, this is about 8 to 15 minutes. So whereas in the not clothing, whereas your bleeding time, that is 2 to 7 minutes, so here it is 8 to 15 minutes is the normal clothing time. Yeah, so this completes the chapter that is the blood, or chapter 2, blood under the physiology. Physiology. This comes under paramedical. Paramedical. That is for the second year students. Second year. Second year. Yeah.